Welcome to our review on producing a genetically engineered organism. So the first thing that we've got here is a quick overview of the process of producing these genetically engineered organisms. And there are three key steps to this. Firstly, we identify the genes that code for our desired characteristic. Secondly, we remove that gene from the donor organism. And finally, we insert the gene into our host organism. Now, the key idea behind producing genetically engineered organisms is that they produce useful things for us. And a key example of this is our genetically engineered bacteria. So we can actually create genetically engineered bacteria that are then able to produce substances that are useful to us. Things like hormones, vaccines, antibiotics. And the process by which we do that is outlined in the diagram at the bottom. So the first thing we do is, from the human DNA, we identify the human insulin gene and we cut it using restriction enzymes. Then, using the exact same restriction enzyme, we then use that to cut the plasmid from a bacterial cell. So what we've now got are two complementary sticky ends. So that when we take our human insulin gene and our plasmid and mix them together with our DNA ligase enzyme, that joins them together perfectly. What we then do is we insert that plasmid into a new bacterium, and that's our vector. The plasmid there is going to be transferring the DNA from one to another organism. Because they're bacteria, what they do is they reproduce by that process of binary fission, so they copy each other and then just produce large numbers, all with the identical DNA, and we grow them in very large numbers in these big vats, and they will produce human insulin, which we can then just section off from the actual bacteria themselves. So we mentioned two key enzymes that are involved in this process. The first are restriction enzymes, and these are the ones that cut the DNA at specific base sequences and leave these things called sticky ends. So when we use that same restriction enzyme on our plasmid DNA, we get the complementary sticky ends. So that means that we can base pair them up later on. The second enzyme that we mentioned was the ligase enzyme, and ligase joins the gene to the plasmid by joining those sticky ends by forming a bond between them. So that then gives us the two key enzymes that are involved in actually making our vector with the gene joined to it. Now, this process isn't perfect. So what we need to do is we've got to check that the gene has been transferred. And the way we do that is by using marker genes. There are two different types of marker gene we can use. We can either use antibiotic resistance genes or fluorescent genes. So if we go through the antibiotic resistant gene first of all, what we do here is we insert our marker gene, so the antibiotic resistance gene, into the plasmid at the same time as our desired characteristic gene. We then transfer that bacteria onto an agar plate that contains the antibiotic, and we incubate it to allow time to grow. Then, when we take it out after incubation, any colonies that are present have the antibiotic resistance gene, which means they also have our desired characteristic gene. So we can then take those colonies and grow them in large numbers. The second example of a marker gene is the fluorescent marker gene. So when we're using a fluorescent marker gene, we still insert that marker at the same time as inserting the gene for our desired characteristic. We grow the bacteria on a standard agar plate, we incubate it, allow time for it to grow, and this time we shine a UV light onto it, and any of the colonies that then glow, as you can see in the picture at the bottom there, have the fluorescent marker gene, which means they've also got the desired characteristic gene. So we can select those glowing colonies, grow them in large quantities, and that means we've got the right desired characteristic gene within all of the bacteria that follow. The key reason for carrying out that check is to make sure that when we're then producing the large numbers of these organisms, that they do have the desired characteristic gene present. So as soon as we've identified them, we can build these large numbers of our transgenic bacteria. Hopefully at the end of this video you can now describe how to genetically engineer an organism. You can explain how bacteria are genetically engineered to produce hormones such as insulin, and you can explain how marker genes are used to identify the bacteria that contain the foreign genes.